For those of you that know me, I'm Oscar Riera-Lizarazu. I'm a faculty member in the Department of Horticultural Sciences. I'm also the department head for graduate programs. Trish Klein is a faculty member in that department as well, and executive associate dean of our college. And of course, you know Dave. So we are the three, PIs behind this effort on genetic analysis and the breeding platform for the Sustainable Roses project. What I have here is a slide that similar slide that Jikan showed you, and this is the constellation of tools that we have through the Tools for Polyploids ACRI project that you've heard about before. I've got a nice little QR code that you can use to get to the website if you'd like to take a peek. And again, it's an ACRI grant, Tools for Polyploids. And what I've highlighted here are 3 boxes for software that we have developed in the project. One is a suite of, or a pipeline management program, if you will, to do alignment, SNP calling, and genotype calling in polyploids. Then Chris has developed also software to determine ploidy of samples based on genotypic data, and also detects aneuploidy as well. And Deacon talked about ViewPoly that was also developed by Chris. So Chris is developing all this software. Deacon is a beta tester and has done a number of software comparisons through this project. And these are the type of tools that are now available for polyploids. If you look at the diploids, we got plenty of tools, but now we've got tools that actually work with polyploid data that can do haplotyping, linkage mapping, QTL analysis, genomic predictions, etc. So these are the type of tools that we'll be integrating into the Sustainable Roses project as well. And if we look at the same graph, but with a more Sustainable Roses, I guess, angle or point of view, we will be genotyping material developed for this project. We've got biparental populations. We'll have rose collections, a diversity panel, and this material will be phenotyped in Texas, Tennessee, and Oklahoma. Texas, black spot cercospora, Tennessee and Oklahoma, mostly rosette disease. And again, we'll take data from all these materials, we'll run them through qploidy to make sure the ploidy is right, we'll make sure there's no aneuploidy, and then we'll run it through our pipelines to eventually do QTL discovery, leading to, of course, an understanding of the genetic basis of these traits, and then also through genomic prediction to look at genome estimated breeding values, which is something I'm going to discuss with a bit more detail later on how and why we'd like to do that, okay? So there's a QTL gene discovery aspect to this project, but then there is also how to apply some of these tools and technologies for our purposes in a plant breeding context. So one of the things that we're doing, and here we have Tessa that you heard earlier, Jikan, you heard of him earlier, and of course, Chris, 2 postdocs in my team, and Tessa is a PhD student, and they've been tasked to develop a GBS platform for tetraploid roses, and we've done a couple of runs to test the procedures. We've got a map from Morton Blush by George Vancouver, which is a population from Minnesota, and University of Wisconsin Riverside are collaborators in this project, and one from Stormy Weather and Bright Eyes. And it looks like we've got a system that is working somewhat. I think the maps for the Stormy Weather Bright Eyes are a little bit inflated, so we need to do a little bit better, I guess, screening of genotyping errors in the data, but otherwise it looks like we've got a GBS platform that is working okay. And that is probably the platform that we'll use for the genotyping for this project as well, okay? All right, you've heard the talks from Jikan and Tessa, and this is basically what QTL discovery looks like to us so far. So we've got at least 5 QTLs identified for rose rosette disease, 12 QTLs for black spot, and 15 QTLs for cercospora leaf spot. So we've got about 32 QTL. So this is great. We have QTLs galore. Now, what do we do with all these QTLs, right? Okay, so somebody will say, yeah, we've got to develop markers and, track them and use them, etc. Let me go over this a little bit so we can think about this a bit more, okay? So on the left side, I've got an example of a QTL carrier, if you will, that has 5 QTL in the simplex configuration, because we're talking about autotetraploid roses, not diploids. And on the bottom, we've got, let's say, a QTL recipient. This is an individual that has no QTL. And if we're going to make that cross to recover another individual that had all the QTL in the simplex configuration again, we would have to use this equation to calculate what would be the minimum number of plants to at least discover one individual with that QTL architecture. And N is the minimum population size, P is the probability of success, and F is the frequency of that particular genotype. And if you plug those values into that equation, you will calculate that the minimum population size would be 145 seedlings. So that's for 5 genes right here. Yeah, it's a little bit over 100. If you're thinking about 10 factors, you're a little bit less than 10,000 plants. And if you're up to 15 loci, you're over 100,000. Okay, this is logarithmic. Okay, so this is one of the issues about what do we do with all this QTL. Okay, all right. There is a way out. There's a way out. All right. And now I'm going to introduce you to what breeding values are. Now, breeding values, believe it or not, were first formulated by Faulkner and McKay in 1983. And the concept is very simple. A breeding value is the value of an individual for breeding, and it is judged by its performance per se and the performance of its relatives. So in this particular example, I can ask you, how do we gauge the value of a bull for milk production if that bull can't produce milk? Well, you look at the performance of its daughters, its mother, or any female relative of that bull, okay? So in this example, this is a sire, these are random dams, and this would be the offspring, and the breeding value really is 2 times the mean of the offspring minus the mean of the herd. And we multiply by 2 because the bull only transmits 50% of its genome to any single dam, okay? Or in any single offspring. So pretty simple. The value of that bull is estimated by the good genes that it transmits to its offspring. In plants, we can do the same thing, is that we can gauge the breeding value of any plant or genotype based on the performance of that plant itself and the performance of its relatives. And the way they do it in animal genetics is they follow pedigrees to do that. In our case, that would be very challenging, not only because pedigrees are not always well described or kept, but because we can make hybridizations between relatives that is impossible in animals, like a granddaughter with a great-great-grandfather, et cetera, and there's a lot of, I guess, inbreeding and et cetera, and sometimes open pollinations, et cetera. But with markers, we can estimate those relationships and follow kind of this sort of an approach, okay? So we can estimate breeding values for plants using markers as well, and we use the markers to gauge the relationship among all the plants. Okay, so this is basically what was done in 2001 for the first time, and this is the seminal paper by Musing et al., folks from Australia, where they actually did the modeling and the mathematics to show that if we were able to estimate the effect of every single marker in the genome with respect to a trait, the sum of all those additive effects was basically the estimated breeding value of that individual. So if you have markers, you can fit the effect of the markers, you sum those effects, the sum of those effects is the breeding value, and that's what this equation means and states. Then what the group in Australia did is then to extrapolate that to a concept that they call genomic selection, whereby if you have these effects calculated and you've got a model developed based on a collection of genotypes and phenotypes, you can then go from genotype to phenotype and you can actually predict a phenotype. So if you have an equation developed for a particular trait, then you can actually predict the phenotype for that trait based on the genotype of an individual. So we can do that for any trait we want. It could be black spot resistance, cercospora resistance, rose-rosette disease resistance, defoliation, plant architecture, flower color, etc. So it applies for any trait. So you can actually develop genomic prediction equations for any trait you want. Now, animal breeders have been using breeding values since 1983. Okay, if you're going to buy good genetics in animals, well, what do you think we buy? You have a herd and you need good genetics. So what do you think you buy in the marketplace? You buy semen from a sire or a bull. And how do you think they gauge the quality of that semen that you're going to buy? Breeding values. So they assign a breeding value to a sire, and the same is assigned to the semen that comes from that sire. Okay, so they've been doing that since 1983, and because of molecular, genomics technology evolution, now they've gone from pedigrees to actually using markers to do the same thing, right? So animal breeders have been doing this since 1983, and plant breeders have started to do this since 2001. And where are we now? 2023. 23 years later, we're talking about this as a new thing, but it's something that has been in the community for a while. It has been modeled, it has been validated, and it works. And it's a technology that is used in large conventional breeding programs in the industry sector. Corn breeders use it all the time. Soybean breeders use it all the time. Wheat breeders are starting to use it. Potato breeders have started to use it. I think there's no reason why we shouldn't use it ourselves. Okay, so again, we can go from genotype to phenotype with genomic prediction technology. Okay, another thing that is important to understand here is what follows. So this is just the platform that we'll use, and GCAN is the one that has been running some of the analysis. And this is the example. Okay, so I have a disease index. And what a disease index is, we score disease for, let's say, multiple diseases like black spot, cercospora, and rose-rosette disease. We have a 0 to 9 scale for black spot and cercospora, 0 to 3 for rose-rosette disease. And what we do is to standardize those values to a mean of 0 and a standard deviation of 1. Then we've got now normalized values for those disease scores, and then we sum across, and that's a disease index. So if that number is low, there's resistance. The number is high, it's susceptibility. So what we did here is to plot number of QTL for multiple diseases and the disease index score. And as expected, the more QTL you have, the more resistance you have for all those 3 diseases. But what if we now overlay genomic prediction over this? So we know how many QTL these individuals have, but now we're going to put their breeding value estimates. And again, the breeding value is based on a GEBV, which is the genomic estimated breeding value for disease index. Okay, so this is an index itself. So what you see is that, yeah, individuals appear as susceptible, individuals here are resistant, and the darker those blocks or squares become is because they have more major QTLs that we've identified through QTL mapping. And if the individuals have a light blue color, those are individuals that don't appear to have QTL based on our discovery efforts, but they seem to be resistance based on breeding values and also performance per se, which means that when we perform QTL analysis, we pick up major effect factors, but oftentimes we overlook minor effect factors as well. And minor effect factors appear to be more numerous than large effect QTL. So when we do QTL studies and we want to breed with QTLs, we're picking some guys that look big, but we're ignoring a lot of little guys. And when we look at them this way, you realize there are some individuals there that would have thrown away based on QTL, I guess, selection, but I would have picked them up if I had used a genomic prediction platform. So I think we need to do both things. We need to map QTL, use them in our genome model predictions, but also make sure that we use a method that is not biased against small effect QTL because they may be necessary, okay? Some of the things that we always say is that QTL mapping underestimates the number of factors that are responsible for the phenotype, and they also overestimate the effect of those factors. Meaning those QTLs that you do in your discovery efforts are not as big as they seem. They only seem big in that particular population. As soon as you move them out, you normally see a reduction in their effect. But genomic prediction is not biased against the small effect loss rate, so you can utilize it. Okay, so that's based on mapping population experiments that GCAN has carried out. Again, another reason why we should maybe use a genomic prediction, I guess, platform in addition to marker-assisted selection, if that's what we want to do. So what GCAN did next was to go back to a collection of varieties for which we had data for black spot, RRD, cercospora, and defoliation. And you see the population sizes in the corners above each of those graphs. And what you see here is the accuracy of the genomic prediction models, and the way that is determined is by looking at a cross-validation test of, you keep 80% of the sample, and then you estimate the other 20% that is not part of your I guess, training population, and you do that 1,000 times, and then you run a regression between the prediction versus the real value. And what you see is that we have accuracies of about 0.42 for black spot, 0.45 for cercospora, 0.33 for defoliation, 0.12 for RRD. Very low, but look at the population size. It's only about 184 individuals for which we have phenotypes and genotypes. Again, when people ask me, how many varieties you want to put in the field? Well, as many as we can, because we need numbers to make this work, because numbers and quality of phenotyping improve the predictability of the models. And if we have models that are very, very accurate, then we can then implement this technology right away. So all of the things I want to show you next are based on this quality of data. So all of the predictions that we've made are, I guess, tentative and, preliminary. But the story that I'm going to tell you is still consistent with what we've seen. Okay, so bear that in mind. We don't have the best prediction models yet, and we're going to be working to make those definitely better. All right, this is one more thing that GCAN did, and it looks like this guy has time to do almost anything. And what he did is we looked at those 460 accessions of rows for which we had genotypic data, about 15 to 20,000 SNPs, and he performed what's called a genetic structure analysis. And what this analysis is a Bayesian approach to use the data, the genotypic data, and estimate how many subpopulations do we have in that collection of accessions. And what we see here is that we have basically three major populations. We have the European chinensis group that Dave talked about earlier. We have the Witcherana multiflora group that was talked about together, and this is the Rugosa Carolina group, okay? So blue, chinensis, green, Witcherana multiflora, red is the Rugosa Carolina group, okay? And then on the left side here, you have basically a phylogenetic tree of all those accessions, and that circle around it with varying amounts of red, green, and blue is basically the amount of the genome for those groups that each of those genotypes have, okay? So that's what that looks like. This is more difficult to interpret than this is. And what we see here is that these are individuals that are fully assigned to either the blue, green, or red populations. And all of the individuals in gray is what we call admixed. They're hybrids, okay? And of course, these guys are hybrids between these two groups. These are hybrids between these two groups. These are hybrids between these two groups, and so forth. So this is another way to look at the data, which I like better than the neat, beautiful phylogenetic tree that GCAN produced as well, and the neat population structure, I guess, circle around it, okay? And GCAN is working to try to publish this, so this is your first view at it. Okay, So what we did next was, okay, so this is the population structure for the 460 varieties. What if we lay over that the top 20 best lines based on a GEV index for that particular trait? So the red dots are individuals that would be considered the best, the top 20% with the best breeding values for black spot, the top 20% best breeding values for Cercospora, RRD, and defoliation. And what you see here, I've put circles around the dots that belong to those three populations that I have on the right. And it's basically the same story that Dave told us about. This will be materials with lots of resistance to black spot, Rugosa carolina group, and the Witcherana multiflora group. Cercospora, we see materials with the European chinensis and the Rugosa. RRD, mostly these two groups, but there's some over here as well. And defoliation, again, all three groups. I lost my other circle over here, okay? All right, so that's one way to see also where are the sources of resistance using a genomic prediction platform, in addition, in combination with a population structural analysis. All right. So this is the population structure. And again, now the same plot as this one, with red are the top performers for multiple traits, in this case, RRD, Cercospora, and black spot. And in blue are all other lines that would be less competitive, if you will. And what we see here is that we've got top performers throughout the span of the diversity of the germplasm, meaning that we have good sources for breeding purposes, if we like. Now, the genomic prediction models are not optimal yet. So I'm not gonna start taking these guys and breed, right? We need to fine-tune those models and redo this analysis, but that's the strategy we would use to start selecting material that's already really good for multiple traits. So with an index, we basically are looking for individuals that have the right balance of traits for rose rosette disease, black spot, Cercospora, and perhaps defoliation, right? So as we saw before, the genetic architecture landscape is complex. There are many genes out there that affect disease resistance. We have seen that there are factors that are positive for one disease, negative for the other. So an index will balance those out. And because we're also exploring small effect QTL in addition to major effect QTL, we should be able to come up with individuals that have the highest frequency of favorable alleles for the combination of traits in your index. And a breeder kind of understands genomic prediction because what we give you is a number, and you can rank genotypes by that number, and you can also apply a breeding index that is, important to you. You can weight different traits according to your preferences, and the index at the end will give you the individuals that have the right combination of traits that you want in your breeding program, whether it's landscape roses or cut flower roses, for example, or something in between, okay? So I think this is something that maybe it's something that breeders and breeding companies can use more readily, because it's like data. It's a value that you can make decisions on, rather than, can I run this marker, can I run that marker, etc. All right? Okay, so what we wanna do is continue to do QTL discovery, and we're going to be using now collections of varieties. So we're going to use association mapping, and we're going to use GWAS-Poly to do that. Again, GWAS-Poly can handle diploid and polyploid data. So we'll use that to do the gene discovery part because we're curious and we're interested in genetic architecture. But then we want to use the stage-wise, which is the genomic prediction engine for polyploids, and we'll use that for genomic prediction and genomic selection research that we're going to be carrying out. We're not going to be breeding yet. We're going to be doing some research and testing the capabilities of this approach first. Genotyping platforms, we have Array, and we are developing GBS. I think we're very close to do that one. And then we're also exploring whole genome sequencing with other collaborators, okay? Again, we're phenotyping RRD in Tennessee and Oklahoma. We'll be doing black spot in Texas, but also Cercospora and most likely defoliation and many other ornamental traits if we can. All right, like I said before, we were interested initially just in gene discovery efforts, but of course we want to apply genomic prediction as well. So our approach is this: identify QTL, understand genetic basis of traits, and let's see if we can implement genomic predictions to identify germplasm for breeding or germplasm for new population development as well. Okay, so that is the new thing that we're trying to do, is do more genomic prediction. So where are we with phenotyping in Texas? And that's something that Tessa's leading and taking over. She does like to go out there and look at the plants, which is great. We've got all these populations that she's phenotyped so far, and these are the number of individuals, but to bear in mind that some of these populations are replicated, so it's not the total number of the population size, but overall she's looking at about 1,300 genotypes. And what she does is to phenotype monthly for Cercospora, black spot, defoliation, flower intensity, and flower number right now. And we'll extend that to other traits when it's time. She's collected tissue for most of the material already, and we were going to start doing some comprehensive DNA extraction and genotyping early next year. I'm getting a new graduate student that hopefully is going to help in that endeavor. So as soon as she gets here, we're going to start doing this. How have we used genomic prediction so far? One of the things that we did do is look at our mapping populations, and we use genomic prediction approaches using trait indices to select 100 top lines from each of those populations in the tetraploid side, and about 50 individuals in our multiparental, diploid populations. Okay, in theory, this could be basis for additional intercrossing and breeding down the line, or germplasm releases, or whatever that is. We don't think we should get into looking at variety collections yet, because again, the genomic predictions models are not optimal yet. There are some genotypes that I have to throw away from those for a number of reasons. But once we have what we call optimal models that have high accuracy, then maybe we need to start even selecting some, I guess, varieties as being especially interesting with respect to multiple traits like Cercospora, black spot, and RRD resistance. Okay. Again, we've got 5 mapping populations, and we are thinking about that 400 variety GWAS that you've heard about. And again, it's for augmenting all of the stuff that we've done, because the more genotypes we have, the more phenotypes we have, the genomic prediction models just get better and better and better, and the prediction accuracies increase as well. And now I need to thank everybody that works with us. And these are the folks at Texas A&M only. Again, graduate students, former graduate students, field staff, researchers, etc. And this would be our collaborators, institution-wise, Tools for Polyploids and Sustainable Roses. Thanks to everyone. There's an incredible synergy that I see happening between these two projects, and we hope to sustain that. We're interested in trying to take the Tools for Polyploids to the next level. This project is going to end in about a year, so we like to renew it. So if you've received a request from me for a letter of support, please do so, because it's going to help the Sustainable Roses project as well, if we keep this moving. All right. And again, thanks to people that sponsored this meeting. And of course, thank you to our collaborators in industry as well. And that's all.